A series of events takes place in cell as cell grows and divides to form two daughter cells. In some of these events, cell spend most of its time there. It grows, it replicates its chromosome and its organelles, accumulates nutrients and prepare for cell division. After that, cell leave that phase and enter into other phase. There it divides to form two new daughter cell. In this way, cell enter into different phase in a sequential manner in a repeated way to maintain its growth and development and sustain its life. This series of events occur in cell is known as cell cycle. Hello friends, I am Suchi. Welcome to my YouTube channel. In this video, we will discuss about cell cycle. In eukaryotic cell, cell cycle having four distinct phases. First one is G1 or first growth phase. Second one is S phase or synthesis phase. Third one is G2 phase or second growth phase, and fourth one is M phase or mitotic phase. All phases occurs in cell in a sequential manner. If we will show, you can represent all phases in this manner. The short duration of a cycle in a particular species is M phase. In between two successive mitotic phase or M phase, there is a longer duration where cell spend most of its time there. It grows, replicates its DNA content, increases its organelles number and accumulate nutrients and preparing itself for the further division. That phase is interphase. This interphase comprises all three phases. First one is G1 phase, second one is S phase, and third one is G2 phases. Activation of each phase depends upon the progression and completion of previous phases. First, we will discuss about this G1 phase. It will start after mitotic phase until the synthetic phase will start. This is also known as post mitotic growth phase. During this phase, cell supplies protein to cell or to organelle. It increases its organelle number like mitochondria, ribosome, endoplasmic reticulum, Golgi apparatus. All organelles increasing during this phase, and biosynthetic activities resume at high rate during this G1 phase. During this G1 phase, at a certain point, cell also decide whether it will continue the cell cycle or it will withdraw from the cell cycle and enter into the G0 phase. This G0 phase also known as resting phase, or you can say quiescent phase. When cell is going for G0 phase, when cell is not getting proper nutrition, or you can say extracellular nutrient or extracellular growth factor is absent in cell, that time cell is going for this G0 phase. During this G1 phase, cell having three options. First option, it will continue this cell cycle by entering into the next phase, which is the S phase. Second option for cell in this phase, it will withdraw itself from this cell cycle and it will enter into this G0 phase. Third option for cell, it will arrest in this phase, either it will once again start this cell cycle or it will enter into this G0 phase. To ensure all these activities, whether cell is getting proper nutrition or not, or cell is replicating its chromosome or not, there is a mechanism to check all this process in this cell cycle. That mechanism is known as cell cycle checkpoint. This checkpoint you will find at certain key step during this cell cycle. During this G1 phase, you will find one checkpoint, that one is G1 S cycling. It is also known as restriction point or start point in each cell. To ensure cell is eligible to enter into the next step or next phase or not. This is the transitional point where cell is deciding to continue the cell cycle or not. This transitional point you will find, or this checkpoint you will find in this transitional stage. Second phase is S phase, which is also known as synthesis phase. It starts when G1 phase will complete and it will end when complete replication process will occur. During this phase, cell replicates its DNA content. And during this phase, transcription process and protein synthesis process rate will be slow. And in animal cell, centriole duplicates its number, or you can say its type. After completion of S phase, G2 phase will start. This G2 phase will start when DNA replication will stop. During this phase, cell grows, protein synthesis will occur at first phase to make cell itself ready to enter the mitotic phase. Here also microtubules reorganize themselves to form spindle fiber. To check in this phase whether cell is eligible to enter into the mitotic phase or not, there is also you will find one checkpoint. That checkpoint is G2M checkpoint. It is controlled by G2M cyclin. G2M cyclin checkpoint regulated by a particular gene, which is a tumor gene. That gene is P53. It ensures whether DNA is perfect or not for this cell division. If DNA is perfect, it will allow DNA to enter into this M phase for cell division, or if DNA is really damaged, then it will allow DNA for its repair, or it will send those DNA for apoptosis process. If by any means this P53 gene is getting damaged in cell, that time cell will grow unlimited and it will form a cancer cell. Another one checkpoint also you will find in this M phase. In M phase you will find third checkpoint, that one is spindle checkpoint. It will ensure whether all spindle fiber is attaching with all chromosome in equatorial plane or not. In this way, cell pass on to different phases and enter into this mitotic phase. There it divides to form two daughter cell. This is all about cell cycle. In next video we will discuss about mitosis process. Thank you.